Thank you for your nice introduction and thank you for inviting me, this your research group and Johns Hopkins. And okay, as Professor Kim, Toko Kim, introduced me, I'm a bioinformatician and I'm mainly developing algorithm for analyzing spatial transcriptomics and single cell analysis, single cell transcriptomics data. And I prepare today two different topics. First one is the same with this title, TENET Plus. It's about analyzing single cell expression and chromatin accessibility data. And yeah, that's this is the first topic. And second, maybe you guys more interested in second topic. It's about cell-cell interaction and spatial transcriptomics. Okay, let's start with introducing my institute. I'm from Sunshine University in South Korea, and I can say this university first modernized university in South Korea, founded on October 10th, 1897, 19th century, and in Pyongyang. It's now in Pyongyang is the capital in now North Korea, by William Baird, a USA missionary. And on 1938, the school closed itself because of Japanese imperialism. And in April 1954, Sungsil was reestablished in Seoul. And so, yeah, this is the brief history. And we started many things in first time in South Korea. We started a computer science department first time in South Korea. And we also started open department of bioinformatics first time in South Korea in 2001. And I was the first student, undergraduate student at that time, 2001, and I came back this university, my university, 20 years later, 2021. Okay, so that's a brief introduction of my institute. And as I said, I'm mainly focusing on developing new tool for single cell and spatial transcriptomics or any other omics data. And I guess you guys already figured out why we need single cell. It's because it's not bulk. I mean, it's not. We can we can analyze the cellular heterogeneity using single cell or spatial data. And I guess I don't need introduce this slide for yeah why we need a single cell. Uh, so as I said, I'm gonna cover two different topics, and whenever I introduce my research, I categorize the purpose of the single cell or spatial transcriptomics into these three categories. And the first one is cellular heterogeneity, and using many machine learning or the PCA or UMAP or the clustering, any method, we can figure out some clusters or the cellular or the sample heterogeneity between different samples. So that's the first topic. So, but I'm not gonna cover this first topic. I'm gonna cover these two different topics. So, second category of the purpose of single cell or spatial transcriptomics is dynamics. So, using single cell or spatial transcriptomics, we can reorganize, we can reestablish the journey of one single cell from one state to different state. For example, if we can obtain the single cell transcriptomics data by different time point, we can reorganize the timeline, assuming we can reconstruct the trajectory of one single cell from one state to different state, and we can also obtain the which cell is represent the middle state of this journey of one single cell. I know this is not single cell. I mean, this is a collection of different cells, and we can say this is just a snapshot of a different state of cells. But we can say, at least we can say, this can mimic the journey of one single cell, then we can also say we can reorganize which genes uh, change first, which genes follow the dynamics of different genes. So we can order the change of the gene expression, and we can also reconstruct the gene regulatory network based on this pseudo-time. If this pseudo-time is if these pseudo-time ordered cells are well represent the dynamics of the biology, then we can reconstruct the genetic network. So that's the first topic I'm gonna cover later. And the third category, the second topic I'm gonna cover is the cell-cell interaction using single cell or this very special sequencing data, the peak sequencing or the spatial transcriptomics data. We can study cell communication, and we can also study which gene is changed by the neighbor information or the cell-cell communication, like using the ligand receptor pairs. That's the third category. So let's cover the first topic. I said the gene network and cellular dynamics. So I guess this picture is already known, very famous picture about the epigenetic landscape. So this landscape represents the potential of cells starting from the stem cell into the differentiated cells, and this landscape is controlled by the gene regulatory network. So this the black part represent the gene, and there is a relationship between genes, and this relationship and gene regulatory network can determine the landscape, and by this landscape we can say there can be canalization of the cellular state from stem cell into the differentiated, or the normal state into the disease state. So I'm going to introduce my tool. I named this tool is TENET. As I said before, if we can reconstruct the trajectory of cells, then we can also calculate the relationship between different genes or different variables. So any measured data from the omics technology, then we can calculate the relationship. And more specifically, we can calculate the also causality, not just the correlation, because we have a time. We can calculate the entropy value from two different conditions. First condition is considering only its own variable, and the second condition is considering both its own okay, this is this green part represent the previous information. So this variable, this entropy represent the condition of its own variable, and this entropy represent the condition of both its own and the other. If there is a big difference between these two entropy, we can say there is a causality between x and y, from x to y. So this is the basic concept of the causality, calculate of causality, not correlation, from the time-dependent data set. So my tool is very simple. So I calculate the transfer entropy for all gene to gene, and only pick up the statistically significant relationship between two variables, and based on that information, I reconstruct the gene regulatory network. Because my tool is based on the transfer entropy, and I name it TENET, but coincidentally, when I developing my tool, one motion picture names with the same name, TENET, is filming at the time. So I very quickly upload my tool into the GitHub to precede this motion picture is released. So this motion picture released on 20 2020, but I upload first before this movie released. So I'm the precede of this movie. Okay, anyway, so in also coincidentally, in this movie, they also talk about the entropy and they also talk about the time. So coincidentally, the topic was very similar with my tool and this movie. Okay, I apply my method to mouse embryonic stem cell differentiated into the neuroprogenitor, and I reconstruct the gene regulatory network, but I didn't show in this slide the very complex gene regulatory relationship. But using my reconstruction of the gene regulatory network, I can suggest which transcription factor is critical. So based on the number of target, based on the network theory, so hop is more important. We can agree about that. Hop more the transcription factor with more target gene could have more important roles in this dynamics of the embryonic stem cell and your progenitor. So when I prioritize the transcription factor based on the number of targets, I found Yamanaka factors, and also the transcription factor is already known, have a very critical role in differentiator of the neuroprogenitor is included in this list. And I compare the capability of finding these already known transcription factors. I compare this capability of finding critical factors with other genetic network reconstruction tool, and I found my tool have the superior performance to finding the critical transcription factors. So every other tools fail to find this Yamanaka factor. Even this factor is very critical on this mouse embryonic stem cell dynamics. So I can publish this in nucleic acid research based on this performance. And I wanna so this is just finding the already known factors, but I wanna apply my method to and identify novel factors using well-known dynamics. So I studied with collaboration with the Seoul National University and Catholic University, and I studied autophagy. So we obtained a single cell RNA sequencing data from this autophagy experiment. This is the just in vitro cell experiment. When we give mouse embryonic fibroblast glucose starvation or amino starvation, they went through the autophagy process. But these two different stimulus can give us two different stimulus can give us the same result, but they may use different pathways. But it's not well known which pathway they use for these two different starvation. So we obtain a single cell RNA sequencing data, and it shows um, the trajectory on this 2D dimension. It starts from this control 0 hour, and it goes through this glucose starvation starved cells and amino acid starved cells, and two different trajectory. And we can say there can be some bifurcation when we give two different stimulus and along with these different time points. So we reconstruct the two different gene network of this glucose starvation and amino acid starvation, and we obtain. We also prioritize the transcription factors, and we found this already known factors is included in this amino acid starvation case gene regulatory network. And we are very curious what is the role of this of uh, the the first ranker, the CBP gamma. Is relatively not known. The role of the CBP gamma is not known in this autophagy process. And we realize this factor is not found in these different gene regulatory network for the glucose starvation. So our question is like this. So is CBP gamma a critical factor for the condition specific, like AAS, the amino acid starvation specific autophagy? So, and we did the experiment and we validated this transfector will really have the condition specific role of this autophagy. So we can say this GFP shows the autophagy process, and when we give amino acid starvation and glucose starvation, in both cases they go through the autophagy. But when we knock down the CPP gamma, only in this case the autophagies go away, but autophagy go away. But in this case, even we knock down the CPP gamma, it's the autophagy is still when we give them the glucose starvation. So yeah, we validate the TENET has a identify the transcriptor, novel transcriptor, and we validate the role of the CPP, the transfectors using this cell experiment. So yeah, after I start my lab, I apply my method to many different cases like liver cancers or uh, the heart fibrosis in using the public cardiac single cell RNA sequencing data. Okay, so from this slide, I'm gonna introduce the topic the same title, the TENET Plus. So I'm gonna just skip this part. So we have many different tools for the analyzing the epigenomic or the different. We have many different omics tools. We can apply them using in the single cell level. So, okay, let's say we have a multiomics data. So we can extend, just to simply extend my strategy in order to find the causal relationship between different variables, different modality. Like some transfector can affect the chromatin accessibility, chromatin accessibility can affect also the target genes, and there can be some triangulated relationship between transcript factors and cis regulatory element and also target gene expression. So very simple, but we collect this triangle TF, CRE, and target gene, and we figure out which transcription factors or which enhancers is very critical for the cellular dynamics. So yeah, this is the simple concept of the TENET plus. We just combine single cell RNA and single cell ATACseq, and as I said, we try to find the causality between transcription factors and target genes and cis regulatory element. And from cis regulatory element, we can also figure out which gene expression is affected by the open chromatin intensity. So yeah, this is the algorithm flow chart, and using some trimming method, we try to trim the false positive, and we extract the triplet, and this triplet shows the relationship between different variable. Okay, so we apply our method to this well-established multiomics data. These data include four different trajectory for the CD4 T cell, CD8 T cell, and okay, this is from PBMC. Okay, CD4, CD8 T cell, and B cell, and monocyte. So we reconstructed four different gene regulatory network, including the cis regulatory element, and we validate the transcription factor and target gene relationship and transcription factor cis regulatory element relationship based on the chip sequencing data. This is the example of the validation. So this is the average transfer entropy, which means the causality of the gold standard, which means from the chip sequencing data, we can collect the gold standard, and this is the average, and this is the the distribution of the null hypothesis. And so we can say this chip sequencing relationship can be reconstructed based on this pseudo-time ordered multiomics data. Also in this case, the transfactor and cis-related element. And we wanted to validate our method. So we have a two different trimming method, and when we applied this, I skipped that. But anyway, so we have two different method, and when we applied these two different trimming method, we can show the transparent values is significantly moved into this the higher values, as you can see in this pink distribution. Okay, that's a validation of the transfer entropy is working based on the chip sequencing data. And this is the second validation. We also wanted to show the TENET plus can also identify critical transcription factors based on the cellular dynamics. So my tool shows the superior performance compared to other tools to identify the already known critical transcript factors. And then this is the third validation. I also wanted to show uh, the causality with the 10 plus suggested is can show the relationship between two different genomic part like enhancer and promoter relationship. So gene expression is globally, gene expression is regulated by also this 3D structure, like because there is a, let's say there is a enhancer, and here is the promoter, and these two different parties have a relationship in the 3D chromatin structure, and this relationship is well represented in the high-C data. So I used the external high-C data, and I wanted to validate the relationship I found from the 10 plus tool. So this is the benchmarking of my hypothesis, and so when I see this the first result, I'm very disappointed, but when I frustrated, but when I see this result, so these two different results shows the based on the distance. So this result obtained from the short distance, and this result is obtained from the very long distance. And my tool can obtain can show the relationship between promoter enhancer enhancer intensity. Okay, so it's very confusing. So sorry. Okay, so I predict so which parties have a relationship like promoter and enhancer, and I collect the intensity from the high C data. And what I mean, when I compare the intensity of the relationship in the high C data, actually there was not that significant changes in this short distance. But when I compare them in this long distance, I can say my tool can figure out which promoter and enhancer have a strong relationship. So yeah, so I also validate the relationship between the promoter and enhancer. And okay, so my tool is working in reconstruct the epigenomic regulation, and also I validated in ChIP-seq and HiC data. And I also wanted to validate 10 plus tool can suggest the novel transcription factors. So this is the prioritized list of the transcription factors. The LLR-FIP1 is the first transcription factor, and I realize all the other tools is failed to find this transfactor is very critical, but only my tool shows this transfactor is very important. So we can say, okay, my tool is very not working, but we can say also only my tool can find this transfactor is important on this CD4 T cell differentiation. So my hypothesis is because the other tools is rely on the motif information. So that's the reason why the other tool is failed to find this risk factor. And this transfect because the reason because maybe there is no well working antibody of this LRIPP1. So chip sequencing is not easy for this LRIPP1. So the motif is not known still. So but my tool is not rely on the motif information. My tool is rely on the pseudo-time ordered trajectory and calculate the causality. So that's why maybe I can find these novel factors. So I validate the role of the LRIPP1 when I knock down these transfectors in the CD4 T cell, and I validate the target gene is followed this knockdown experiment. And I also found the LRIPP1 knockdown experiment shows the reduced terminally divided T cell proliferation. Actually, I failed to find that this knockdown effect is knockdown, this LRPM knockdown have a role in the differentiation. But anyway, so I found that this knockdown have a role in the proliferation of the CD4 T cell, and maybe this LRP1 is very critical for the role of the CD4 T cell in the immune response. Okay, so I'm gonna skip this. So that's the first topic. So using transfer entropy, I'm applying this causality tool to the many different the modality, and also currently I also apply this method to the spatial transcriptomics. Okay, so I'm gonna move on to the next topic. Using single cell or the some spatial sequencing data like two cell data or the spatial transcriptomics, we can figure out the cellular communication. So the conventional way of studying the cellular communication is actually using the database. In the database, there is a already known relationship between ligand and receptor, and using that information, we can figure out, we can calculate we can identify the significant relationship between two different cell types. If one cell type have a one ligand and the other cell type have the receptor of the partner, then we can say these two cells have information the cell-cell interaction. Yeah, that's it. That's the conventional way, but it's not perfect. So it's just a guess, and this is it's just based on the database. And in our group, we try to suggest the cell-cell interaction not based on not based on the database or the line receptor information. And there can be also novel gene pairs between two different cell types, not like ligand receptor pairs. Okay, that's the our concept, and I'm gonna introduce two different tools. And first part is based on the two cell data, and so we call this physically interacting cell sequencing, aka two cell sequencing, because it's not easy to figure out, as I said, using only database the which gene have an interaction, but when we can obtain two cells, we can say, okay, there is a interaction between two cells. Okay, so the question is like this. So are these two cells in the same state? So these two cells are blue cell, which means these two cells have the same markers, which means these two cells are same cell types. Or let's say in the pancreas, there is a beta cell. Okay, this is beta cell, and this is beta cell. And let's say this beta cell surrounded by beta cell. But this beta cell, let's say this beta cell is surrounded by alpha cell, then these two cells can have a different state, which means different gene expression. So we can imagine like that. But it's not easy. So which gene was surrounded by the same cell type? Which gene is surrounded by enemies or different cell types? So, but if we can obtain two cell at the same time, and sequencing them, and we can compare the expression difference. Then we can figure out which gene is affected by different neighbors. That's our concept. So, we obtain, so we apply our method to this peak sequencing data of mouse embryo, 7.5, 8.5, 9.5. There is a very dynamic changes in the interaction between different cell types like endodome or neuroprogenitor or like these different cell types. So we obtain single cell and two cells from the same experimental not the same sample, but yeah, the same experiment. And is there any chatting? Okay. So then we can compare these two. And but we need some artificial computation, like because in the single cell, there can be also mixed up of the these two different cell types, same cell type but different state. But anyway, in this single cell RNA sequencing, we can say it's like null hypothesis, and make based on the artificial two cells, we can make the null hypothesis, and we can say that's a reference, and we can compare this real two cell and artificial two cell. We can say there is a the gene, we can find the gene affected by the neighbor information. Okay, so it could be very complex concept, but anyway, so we have a, let's say we have a two cell and we have a single cell. As you can see, these neuroprogenitor have their own marker, and endodome also have their own markers, and we can statistically identify the markers for unregulated when only these two cells are have a neighbor each other. Then we can find these genes by comparing the single cell and the real two cell. So we can say these genes are unregulated when these two different cell types are the neighbor each other. So we validate the gene expression using spatial data, and also this cell experiment. We obtain the endodome and progenitor, and we also so we give the GFP. I'm not experimental guy anyway, so we give a GFP and we co-culture this endodome and progenitor, neuroprogenitor, and we can also using FACS, we can separate these endodome and progenitor was co-cultured each other, or the how can I say, monocultured itself, like the endodome and the progenitor. And these are the genes I found in the two cell data, and we validate these genes are really unregulated when only these two different cell types are co-cultured in this experiment. But it's not sustained in different in the along with this time point, this gene expression is came back to the normal expression levels like this. Okay, that's so we can publish this based on this peak sequencing data and computational method, and also validation. We can publish this in PNAS. So we also extend our concept into the spatial transcriptomics. So because in the spatial transcriptomics there is no dissociation. Uh, the how can I say, main shortage, or how can I say, main point, shortage. Yeah. Anyway, so in the single cell data is because we need dissociation in the single cell RNA sequencing data. But if we can obtain in situ the transcriptomics in the intact the tissues, then we don't need to the dissociation. Dissociation can also affect the gene expression, and also dissociation can lose some information of the neighbor interactions. So we have a spatial transcriptomics. So and we have a concept of the figure out which gene is affected by the different the neighbors. So we apply our concept into this spatial transcriptomics. And he published in Molecular Systems Biology 2023. And we named the tool name as a Cell Neighbor EX. And this tool provides a new perspective on cellular communication between adjacent cells. Okay, so the concept is very similar with okay, concept is very similar. So we have a spatial transcriptomics data, and we can also obtain a reference single cell RNA sequencing data, and we can deconvolute the spatial transcriptomics data. But or if it is image based, we don't need deconvolution actually, and we can obtain the which cell type is have a neighbor each other. Okay, so based on this, we also reconstruct the artificial heterotypic spots, which means artificial two-cell data. It's same with the previous concept, and we can compare the gene expression between this artificial data and the heterotypic spots of consisting the two different cell types. Okay, that's a concept. So the artificial heterotypic spots, and we can also obtain the real true heterotypic spot or bead, and we can compare these two cases and figure out which gene is affected by the cell-cell interaction. So yeah, so forget about this. So it's image based. So it's a different strategy. So more easy strategy. But anyway, so we have if we have an NGS based spatial transcriptomics data, then we can compare the heterotypic spots with these homotypic spots, and we can figure out which gene is unregulated by comparing these hetero and homo cases. So this is the also validation. So cell neighbor EX found 155 unregulated genes from 21 heterotypic and pairs, and 55 downregulated genes from 8 heterotypic pairs based on this mouse brain data of slide C. And this is one example. FABP7 expression in slide C data is highly expressed when only these endocellular tip and astrocyte are neighbor each other in this spatial terms. And we also validate the expression changes using this co-culture system.